Hi again, and welcome to part 2 of visualizing gene expression data using interactive clustergrams built with D3.js. In this part, I'll be discussing network visualizations and clustergrams. So, network visualization is a common task in many fields, and often networks are visualized as nodes and links, which is what we could see here. So D3 has a force-directed graph layout for this type of visualization. And in this visualization, you are seeing characters from the book Les Miserables, and they're connected based on co-occurrence in book chapters. So this data was manually curated and available on the Stanford GraphBase, and it's a database or data set that we'll be referring to a few times during this part of the lecture. So if we follow this link, we can see a live example of a visualization of this network. So one of the most used components of D3 is this force-directed graph layout. So you can actually move the graph components, and the components are linked to one another through simulated springs, and you have basically each of these, the force at any given time is calculated using a physics inspired algorithm, and you have repulsion occurring between all nodes, and the nodes are attracted to each other based on some degree of gravity and also based on the spring connectivity between these nodes. So what this allows you to see is how these characters and these nodes cluster together and form these different clusters. So they're actually colored differently, so it kind of helps you highlight what's going on. And you can see how certain characters co-occur with each other, and some of these all co-occur with this character, and then these, by comparison, all co-occur with each other. So you get some idea of the interconnectivity and the structure to this character network. And if you hover over each node, or any node, you can see the character that is actually in this node. This node is representing, and the thickness of the link represents the frequency of co-occurrence in different chapters. And then down here you can see how to use this layout, so it's not that many lines of code, and you can see here the data structure for representing this network, and it's a data structure that you'll be seeing a lot more of. It's a JSON structure, so it's a way of storing data. So this network visualization is very popular, but probably the main problem with this type of visualization is that when you're viewing very large networks, it often results that you get a large hairball, which is very difficult to comprehend and interactively explore at all. So this is an example of a large hairball network from this paper here, and I believe they're viewing genes, and what it's showing you is that you can see there's some clustering going on, and they've colored these nodes to some degree, or the links to show a type of clustering, but it's very difficult to get any real idea of what's happening. And if this is a static picture, you pretty much can't gather anything very meaningful from it. And this is a problem that exists with these types of networks. But alternatively, networks can be visualized in what's referred to as an adjacency matrix or a similarity matrix. So in this case, we're viewing the same character network from Les Miserables, and the characters are this time shown as rows and columns of a matrix. So this approach has the advantage of being able to visualize larger networks without forming a hairball. And this is because you never have any overlapping links, in that each link here is no longer a line, but it's a tile or a square in this matrix. So you can see, if we go to the interactive example, some interesting features that are sort of D3 specific. So here we can see the data in a matrix form, and one of the things this example is trying to make very clear is that with this type of network visualization, it's very important to order your nodes, in this case, properly. So by changing the order from name, which it was the initial order it was in, you see very little structure. So you can see which characters are linked to one another, and if you hover over them, it highlights the two characters that are connected. And the opacity gives you an idea of the degree of connectivity. So the number of the darker this square is, the more chapters the characters co-occur in. But you can also reorder this matrix based on frequency, so you can see characters that just very frequently co-occur with each other and characters that don't co-occur with each other. And once again, if we put it in cluster, then we can see the clusters that form, and these clusters are colored, are in different colors. So this type of visualization has the advantage of sort of over the visual space spreading out its information and preventing us from getting any sort of line crossings like we saw with this equivalent visualization. So here you can't really tell exactly what's going on because there's a lot of lines crossing, but here you don't have that problem, but the visualization is, in a sense, larger and more spread out. So there are two ways of viewing the same data. And a similar visualization called a clustergram, or a heat map, is a visualization that is designed to show clusters in your data. So here it's similar to the similarity matrix or adjacency matrix, except that now the rows and columns are no longer the same. So in this example visualization from this link here, it's a heat map drawn from the programming language R. And what we're viewing here are car models like Honda Civic, Toyota Corolla, and we're comparing these models based on their attributes. In this case, it's gears, miles per gallon, cylinders, this kind of thing. So you can see which cars are similar to one another and what attributes make them similar. And you can also see which attributes are similar to one another based on which cars they occur in. And here, this dendrogram view allows you to see a hierarchical tree of relationships among these. So you can see how these cars sort of are in two large clusters here, and then there's smaller subclusters that occur through this dendrogram. So in this visualization, the rows and columns are generally not the same. If they're the same, then you have a symmetrical matrix, and you have an adjacency matrix. So I'll explain the relationship between the previous visualization and this clustergram visualization next. And so shown here are the two visualizations using a ball and stick network and a similarity matrix. So these two visualizations are showing the character co-occurrence, and a clustergram can really be thought of as an extended view of either of these similarity networks, in the sense that each of these networks is showing you the network structure of the characters. So we can see clusters of characters occurring. Like here we can see a cluster, here we can see a cluster. But what these visualizations don't show you is the evidence that actual components that are similar among these characters. So the actual book chapters that they co-occur in are not shown here, so they're sort of hidden. They are only represented minimally in this case the width of the link, and in this case the opacity of the square. So on the other hand, a clustergram really allows you to have an extended view of a network, and it's in certain cases where in this case we have our characters shown here as columns and our chapters shown here as rows. So now we can see which characters are similar to one another, and we can see which chapters they co-occur in. So we're effectively, with a clustergram, viewing two networks simultaneously. We're viewing the character network. We can see which characters are similar to one another, and we're also viewing the chapter network. We can see which chapters are similar to which other chapters based on character occurrence. So a clustergram is sort of more information dense in many cases. And often clustergrams are used to visualize gene expression data. And these clustergrams can be used to find clusters of genes, in this case, rows, or samples. So in this case, we're viewing differentially expressed genes as rows here, and we're viewing samples as columns. And that's it for this lecture. Next lecture, I'll be discussing how and why we are building a clustergram visualization in D3, and some of the unique advantages that are associated with this. So thank you for your attention.